Matt was giving me a hard time about.
Yeah, well, it's very nice.
We're not trying to vouch for it, I keep overlapping. It's my team.
Good afternoon. We're going to get started. I'm Dr. Kay Stefanik, Assistant Director of the Iowa Nutrient Research Center. I want to welcome you here all to our INRC Spring Seminar Series, Focus on the Future. Today we have two graduate students, both from Iowa State University, who are going to be presenting on their work on INRC funded research. And hopefully they'll talk a little bit, not just about their research, but also as, their experiences as early career researchers. I have a few housekeeping items before we get started. The first is this seminar will be recorded and it will be posted online to the INRC's website, hopefully by next week. And then the second item has to do with questions. So we're going to have a question and answer session after the presentations. If there's time after the first one, we might have maybe a couple questions, but we'll definitely have time at the end. If you are in person, just raise your hand. If you are online, you can either enter your question in the chat box and I will read it out, or you can wait until we're at the Q&A session, unmute yourself and ask your question directly. So our first speaker today is Arturo Flores, who's born and raised in Guatemala and is a master's student of the Soil Science program at Iowa State University. He works at the Geospatial Laboratory of Soil Informatics under Dr. Bradley Miller's advising. So before ISU, Arturo got his undergraduate degree in agricultural sciences from Earth University in Costa Rica, working with tropical crops and precision agriculture. His current research is looking at the spatiotemporal variability of soil in different mapping methodologies. So, floor is yours.
Thank you. Thank you for that introduction. Hello. Well, as you can see, my name is Arturo, and I'm going to be presenting a little bit of.
I want to be able to, okay, I can pick up the mic.
The mic is that way.
All right, I'll try to stay around here. Okay, my title for the presentation today is just spatiotemporal variability of soil fertility with digital soil mapping. I'm gonna talk a little bit more of what everything here means. So I forgot we had to send an introduction, but I had something briefly here. So I was born in Guatemala, I went to Costa Rica, I learned about agriculture. I was born and raised in an agricultural family. So all my life I have been involved in farming, basically. I got my degree in agricultural sciences. I got interested in soils and irrigation with some precision ag. I work in a golf course. I was mowing grass. That's funny, but I was mowing grass. And then I came here to work with machine learning. So that was like a big shift in my life. And now I'm working with Dr. Bradley Miller here under his advising in Geospatial Laboratory of Soil Informatics. But yeah, it's just a brief introduction. So I think we're here because we want to somewhat talk about nutrients or pollution or soil or all of these combined together. So what we want to do is like monitor soil nutrients. That's like our big main scope here. We can see this from two perspectives. First, we have the agricultural one. We know that soil nutrients are important for yield. Like if we have too low of a nutrient, this is going to limit our total yield. That's going to mean economic losses. But if we over apply to reach like certain level in our soil, that's also mean losses because that doesn't translate into more yield, right? And this affects environment. Like if we put more than what we need, it somewhat ends somewhere, especially in watersheds or water bodies at some point. I have like this little model of a soil. So everything in the soil moves, transforms. It comes from artificial products or synthetic products. It can go in and out in any direction, down, up. So it's just a complex system we have to consider. And how can we monitor those changes or those concentrations we have? We're going to cite here Lord Kelvin. So this quote, if you cannot measure it, you cannot improve it. The only way to know what we have in our soil is go out there and take a soil sample. So how do we do that? There's this guide, how to take a good soil sample, make good decisions. That's from Iowa State, the Extensionism and Outreach department. So they state some guidelines, more focus on the agricultural side, like how to help farmers take their soil samples. And they say that if you're doing grid sampling, like your minimum density should be like 1 sample per every hectare or 2.5 acres. You should take surface soil samples like the top 15 centimeters and composite samples. That means like 10 or 12 cores of soil around your location to get like a homogeneous mixture of soil and a broader perspective of what you have there. But this also comes with limitations, like the number of samples. We know that we cannot take all the samples we want. The more we take, the more we have to analyze. It means more money. It doesn't really translate into more economic income, so not everyone does that. And also the time it takes to take more soil samples. Another limitation is the accessibility. Like if we are talking in a field scale, it's easy to just walk our field and take samples, but if we talk in a watershed scale, we cannot go everywhere. Accessibility is not that easy, but also technology. Even if we get a lot of samples done, there's not everyone has the knowledge to analyze that information, or they don't have the tools to analyze them. So what's information without interpretation is just useless data. So we end up with having to use like scarce or dispersed in space observations, and then some kind of modeling to try to predict where we cannot access or sample. So that's just a little bit of introduction. So what we do for our study here, we took a field in central Iowa. That's Boone County, Story County. So it's like 15 minutes away west from here. It's called the Sorenson Field. It's part of the research farms from agronomy and engineering farms. It's a 15-hectare field that we divided into 100 individual points. Like each one of these points is one soil sample. We sample this every 28 days, like 4 weeks in between, starting in March until October. So we could capture like that temporal change on soil. So the samples were individual samples rather than composite, as the general guideline said, but we wanted to have like an absolute location on soil, so we could then do some machine learning with this data. We took surface samples and analyzed them at Midwest Labs. And then, well, we took another guide. It's called the General Guide for Crop Nutrient and Limestone. So this is also a guide from Iowa State. It just tells farmers their sufficiency levels based on their soil results. So they take their samples, they look at this, and they can say, okay, my soils are below a threshold, I should apply this amount of fertilizer. So it's just a way to know how well their soils are according to concentration. So this is just a temporal animation of our soil samples. Each one of these squares is like one soil sample representing each nutrient. So we have nitrogen, phosphorus, and potassium. And then these are the colors. Red and yellow are low or very low. Then the light green is like the optimum, and dark green will be like high levels. So if we take nitrogen, starting in, let's see, now in April, we see how generally levels increase, and then in July, they kind of decrease, like in a broad perspective. Phosphorus, we can see that it's changing across space and time, but there's not really like a really easy pattern to see here. And potassium is like the same, like we have some changes, but not like really easy to identify. So this helps us see that, okay, things are changing with time. But what would happen if we take like the general guidelines that they recommend, like we would reduce the resolution or make samples bigger. So we group those per 2.5 acres, and we end up with these kind of maps. Like we can still see things are moving, but the level of detail they're providing is like really coarse. We are losing a lot of detail here, so we don't really know where things are moving or how much in case they are moving. So this leads us to the objective of this study. The main objective, we divided this in phases. So last year was like the exploratory phase, see what was going on in our field. And we wanted to know the nutrient variability across this field. We mainly want to see if there was a temporal and spatial behavior, like if this is changing over time, is this changing over space, what is happening? Then we wanted to see if there was difference, like if we take different parts of the field, not like the whole field as a unit, and then just create some spatial distribution maps, because that's the way we communicate spatial data, right? There is a couple projects going on here. I think they're gonna talk about that in the next seminar. So there were like 4 big plots, but in between the plots, they were trying some nutrient or nitrogen rates. I think I won't get into detail because I don't really know what Mila was doing there. They were trying to capture different landscape positions. They were trying some pattern of planting here and fertilizer rates as well as here. And generally, they had 2 nitrogen fertilizers applied, like outside of Mila's plots. They had like ammonia here and a mix of fertilizers in the southeast corner. So those were like outside of our project. They were just occurring there. We were just sampling everything and trying to capture as much data as possible. Then we got our digital elevation model. This is just a representation of the topography. So here in the low southwest, we have the high spots, and then the green ones are the lowest. So we have like a decreasing elevation here. It's just a relative change of 9 meters, so it's not like a big deal, but it's still a significant slope. So we use a model that is, well, it was proposed by Miller and Schatzel. They just classified hill slope position in 5. We have our summit in the top and our shoulder that are like our erosion areas. Then we have a back slope where the slope like goes down. That's just like a transport region. And then we have a foot slope and a toe slope where materials tend to accumulate more. And we end up with this map, like blue and light blue. Those are our foot slope and toe slope. Then our white one is our back slope, and this is our summit and shoulders. So we can see that we have like 2 hills and then 2 low areas, like generally looking at the field. All right, now we can talk a little bit of what we did with all the data. So we had 100 points analyzed every month. It was just a huge database. In Midwest Lab, they give you like a report, so it's just a lot of cells to analyze. But we wanted to see what we have here. So we did a descriptive statistics just to get an idea of how nutrients were behaving. We did this treating the field as a whole, but also as at different hill slope positions to see like what is going on in each hill slope position. And we're gonna be focusing a little bit more in nitrogen and phosphorus. So these graphs kind of capture the field level, like field scale level. So we kind of see that nutrients in March, well, this is nitrogen, it starts increasing until we reach a peak in June and then significantly decrease and somewhat remains constant. Then we see there's similar pattern here for phosphorus. Also in June, we reach like a peak and then decreases, but not that much. But we can see that there's a lot of variability, especially for phosphorus. We have some trend lines here. So we wanted to know if these changes are significant, right? What is it telling us if it's not significant? So we run a t-test just to know if the increase we are seeing was significant from month to month or the decrease was significant. And these dashed lines, that means they are not significant. And the color lines, they mean there's a significant change. So we can see there's like a significant increase in nitrogen from March to May. Then it somewhat stays constant, and then a drastic decrease after June. And then it just remains constant for the rest of the season. And then in phosphorus, we see like the same pattern, not as marked as here, but it kind of increases. We have a slow decrease and then somewhat remains constant. But we have to remember that there was something going on here. There was a crop. So it's not that nutrients are just disappearing. So it was in between April and May, they planted, then crop or corn starts growing. We have B6 stage like mid-June. And we can see like that decrease stops when we get to B18, like vegetative growth kind of is over there. It's not like they are not getting more nutrients, but most of their vegetative growth already occurs. So that coincides here with the decreasing nutrients. And then it somewhat remains constant for the rest of the season until they harvest in October. Also, it's important to mention here that they also apply fertilizer. So those increases are not like magic of nature. we have to put some fertilizer in. So that's kind of related to those increases. Okay, so with this, we can know that the temporal effect is like significant. There's something going on over time. But what is going on in space, like topography, has something to do or not? So we took our Hilo position, our soil samples, we grouped them by Hilo position or by region, and we calculated the same statistics for each of those. As you see, it starts getting bigger and bigger, the database. So we just summarize it in, I hope, easier to interpret plots. So, okay, here we have the shoulder, summit, back slope, toe slope, and foot slope. And we see the general trend that we saw before, like an increase, decrease, remains constant. Increase, kind of decreases, and somewhat remains constant. Here for nitrogen, it's not that hard to see that in March and after June, the variation in any hill slope position is like not that big of a deal as compared to during April, May, and June. Like we have a lot of variability. We would expect the low areas to be collecting or capturing if things are moving on space, but we don't really know. But we know that the toe slopes and the foot slopes have a lot of variability. And in nitrogen, they don't have like a higher concentration. But if we look at phosphorus, the low areas tend to have like all the time higher variability and also like a higher mean in between that area. So now we know there's a change on time. We know that hill slope or topography is having some effect here, but what about the spatial movement or spatial data? So we use some geostatistics. We use something called the variogram. So we plot, well, the units here don't really matter. We're just going to be talking about what this is used for. So we plot how, like, the difference that exists is the variance that exists among points at certain lag distances. Like here, we're going to have our distance. So as distance increases, this model is showing us that there's more like variability compared to when they are closer to each other. What is important to take out of here is that there's a nugget effect, or it's also like a measure of the error in our measurements, or it's a just indicator of natural variability of soil. Then we have a seal, is where that model that we feed it into our data reaches like a plateau. That tried to explain a little bit of the maximum variability that we can capture here. And then we get a range, like the maximum distance at which we can get some predictions of independence among our samples. But then we calculated a ratio that is called the nugget to zero ratio. It's just to explain like this effect compared to our whole variance. And the smaller this gets, it means that we can explain that variability like a shorter distance. But the flatter our model gets is like just random occurring. We cannot really use it to predict something. So we calculated a spatial dependence. Strong is below 25%, and weak spatial dependence is above 75, and anything in between just means that they are moderately spatially dependent. So we took 3 dates, April, June, and October, like 3 critical stages. Our initial, well, the start of the season, our peak nutrient levels, and then after we harvest, like what we want to see, how much we have in our soil. So in April and June, nitrogen and phosphorus have like moderate spatial dependence, so they are somewhat spatially dependent. But in October, we see that they are strongly spatial dependent. So what can we do with this? This suggests that distance has like a relation between those. They're related. So we use kriging. That's like an interpolation technique based on distance. The closer they are, the more similar they are going to be. And then we created these models. So here we're including the temporal and the spatial variability of our soils. We have phosphorus here. We don't see like those big changes, but we can see some patterns going on and some hotspots. And then in nitrogen, I think it's easier to see how we start with a low concentration, increases until June, then it decreases. But we can still see like some hotspots occurring until September that it's almost the same level everywhere. Okay, we have some conclusions from our first phase or exploration phase. As we would expect, like the nutrient concentration is varying across landscape. This is like nothing new, but we know it's occurring here, but we have to know that it's significant. So we have that. We also know that there's a temporal effect. We know now that things are changing over time. We don't know what's causing those variability or those changes, but we know they are occurring. And if we want to increase our sampling density, this will be more expensive. So sampling is still like a limitation if we want to get a higher resolution on our data. So what if we look into the future? What if we want to upscale this into like a watershed level? Well, we can still do that, but we have to change to include more digital soil mapping techniques like machine learning instead of using geostatistics, because geostatistics are like based locally, so we cannot like extrapolate beyond our study area. But with machine learning, we can make that association, like we can see what's going on here, and through different algorithms try to use information from other sites and try to relate them. So that's like a opportunity we have here. And now that we know things are changing on time, we have how they behave month to month. We are like thinking, can we make temporal predictions by knowing what we have at the start of our season, knowing how nutrient kind of behaves through the season? So those are just some ideas we have for the future and some references in case you want to read a little bit more. So that's what I had to share with you today. Any questions, feedback?
We should have time for maybe one or two questions before our next speaker. Are there any questions in the room?
Is this the field that we're seeing there?
Yeah, this
Okay,
is the field.
you know, there's not a lot of topography, but you're getting different.
We are here in the northeast, and the highest point is like somewhere here. So you can still see, well, not like a big change, but the color looks darker. So when I was sampling, I was taking all the samples like myself, so I could walk all the field, and you could see like down here it was always wet. So there's like a big change here, and if you go up, I used to find like more sandy soils or more stones. So yeah, there's like a big difference. Yeah.
What year was your sampling done?
Yeah, I forgot to mention that. That was last year, 2023. We started in March, and we finished sampling in October.
When were the samples taken in the month? Like, so when you said July, like that really low nitrate, when were those samples?
They were taken, I have the specific dates, I don't put them here, but they were taken in between the 20 and the 30 of each month.
Okay, so toward the end of
the month.
Do you think that would look different if, per se, it was a normal precipitation year versus a wet year?
Yeah, I think that could have some impact. I found a couple research done in, I think it was Idaho, but it was done with wheat, and they were evaluating that, like, according to precipitation, they found some movement of nutrients downhill. We didn't evaluate that here, but more rain could mean more hydrologic process going on, so that could impact our nutrient distribution.
So, what the yield map looked like, followed up on Mark's question, was there a yield impact?
Yeah, I have the yield data, but I have like individual points, so I don't have anything to compare it with. But it kind of follows the pattern of the topography. Like we have the highest yielding points, not like in the lowlands, but kind of in between the highlands and the lowlands. I have all that data. I didn't put it here because I don't really know how to relate it now.
Right, so we're gonna move on to our next speaker, and hopefully we'll have a little bit of time at the end where we may have just gotten a question online where both of our speakers can have a chance to answer some additional questions. Okay. Right, so our second speaker is Mila Vicorio Pessoto, and she's working on her PhD in the Department of Agronomy at Iowa State University with a focus on crop production and physiology. Her main advisor is Dr. Mark Licht, and co-advisor is Dr. Marshall McDaniel. She's working on research to better understand nutrient, pathology, and agronomy-related causes of corn yield drag following a cereal rye cover crop.
Thank you for the introduction. First, Mark, can you hear me? I'm a little bit soft. You can hear me higher? Okay.
Not really loud,
What?
not really loud.
Yeah, I'm gonna start yelling here.
Yelling at you.
So, as you said, I'm Mila Pessoto. I don't have introduction slide on myself, but I'm from Brazil. I did my undergrad at the University of São Paulo in agronomy, and I got my master's at University of Arkansas in soil fertility. I moved to Iowa to work on my PhD in 2022, and I've been enjoying the winter here since. So my project, the title of my project and the title of the project that we applied for the INRC is Can Adjustment to nitrogen rates reduce corn yield drag and disease implication following a CRI cover crop? So first I want to acknowledge and thank Dr. McDaniel, Dr. Lake, Dr. Robertson, and Dr. O'Brien for all the help. They're all the faculties that are involved on this project. So first, that's not the one. Okay. Okay, it worked now. Oh, thank you. So a brief introduction on cover crop and CRI. The USDA just released the latest census on agricultural census, and it says that the US has 8 million acres on cover crops, and a little bit over half of that is here in the Midwest. So the Midwest has 10 million acres on cover crops, and between all the states, between all the Midwest states, Iowa is the state with the most cover crop acres, with 1.3 million acres. And in Iowa, we have CRI as our primary winter cover crop because it's the most consistent in establishment and overwintering. So most of our farmers here choose to use CRI even before corn. Okay, so CRI can bring several different benefits to the environment and to the following crop. Some of the primary ones are to produce large amount of biomass, which is great for weed suppression and erosion control. They have deep roots, which also helps with erosion control and decrease soil compaction. They have some allopetrochemicals that can be used for weed suppression. And another characteristic that CRI has, it has a high CN ratio around the time of we terminate CRI before corn. So that CN ratio is going to be 42:1. So as I point out, CRI has a great benefits to environment and to the following crop, but some recent studies have been shown that CRI can decrease corn yield. So I have a picture that I took from a publication that came out to 2022 that shows the yield impact on the corn yield impact when following any cover crop. This paper doesn't focus on any specific cover crop. It just has cover crop in general. But I'm getting ahead of myself. Sorry, let me get back a little bit. So on the figure, we can see that the darker the red, the higher the yield impact. So I don't know if everybody can see, but on the scale here, the darker hair, it means that we have up to 10% yield decrease on corn following a cover crop. So, and as I point out on the previous slide, in Iowa, the main cover crop is CRI. So we can somewhat conclude that this yield decrease is being caused by CRI. So that brought us to our question is, does CRI has any harmful effects on corn? And those are the questions that led us to this project and the application of it. So hypothesis that we have that we're trying to understand is if CRI causes a green bridge effect for seedling disease, which is PTM. I'm not going to talk much about PTM on this presentation because that part of the research is being led by Dr. Robertson on the pathology department. I'm focusing more on the nitrogen side. So the second hypothesis is that the CRI is affecting the nitrogen mineralization on that soil. And the third one is that since CRI has an allelopathic effect, those chemicals are hurting the corn on the early stages. So those are the questions we're trying to answer on this project. And to answer that, we came up with this project. The main location of this project is Ames, Iowa, with Central Iowa. We have data from 2022 and 2023. We were renewed for 30 years, so thank you for that. So we're gonna have another one, another field until to 2024, which we're gonna do all the same measurements. Our project 2022 and this year, it happened on the Cali farm, and 2023 was on the Swanson farm, which was the same farm Arturo was talking to us about. We have our project was replicated on 3 different landscape positions on the summit, backslope, and toe slope, because we wanted to see how the landscape would affect since we'd have different soil gradients. And the maize was on top of that, the corn was planted on a split plot design. So the main plot of the split plot was the CRI. So we had a plot with and without CRI. Those CRIs were planted on fall right after soybean harvest. And it was early October following recommendations, IS state recommendations for cover crop planting. And the subplots, we have 3 different nitrogen rates ranging from 0 to 336 kilograms per hectare. So throughout the presentation, I'm going to be using metric. I apologize if I switch to English units by mistake, and we have 6 different units. So as I mentioned, the main focus of this research is to track the nitrogen mineralization. So for that, we did 7 different soil samplings throughout the whole season. So the first one being right before planting, and we had 3 until V6, and the other 3 was every month until pre-harvest. So the soil samplings only happen in 3 different nitrogen treatments. The 0 and the 336 was chosen because it was the highest and the lowest one, and we chose 202 as our third nitrogen rate to be sampling because it's the closest of what the farmers would apply to their fields. So to do the mineralization part, we do a composite samples of a composite soil sample of our plot. This composite sample is divided in 2. The first sample is going to be called sample 0, and that goes straight to the lab. So we sample and we send to the lab for analysis. The second one, we fill a plastic tube and we bury it back in the soil. So we're using a tube, but it's similar to the buried back method that's more common. And it stays buried for 14 days. After these 14 days, we're gonna unbury them and send that soil to the lab. Both samples are gonna be analyzed for nitrate and ammonia. So here I have a picture on how the tube is buried on the soil. You can see that it's all the way down. So we left the top out just to see where it was, but doesn't have any soil interference. Everything is buried and under the soil. Okay, and so to analyze nitrate and ammonia, we just use a standard colorimetric methods. The right one, my right, your left, it's the pink one, that's nitrate, and the yellow one, it's yellow-green one, that's ammonia. And using this method, the darker the color, the more nutrient that sample had. So to calculate the net nitrogen mineralization, we just do a simple calculation where we take the results from the sample 14 minus the results of the sample 0, and that one minus the other, that means that's all the mineralization or immobilization that was happening on that soil at that time. So now moving to some of my results. So on this graph, I have day of the year on the x-axis, and I have net nitrogen mineralization in milligrams of nitrogen per kilogram of soil per day on the y-axis. I have all three different nitrogen rates and all three different landscape positions. So before I explain, so another thing to point out is we have a dashed line on the 0, and everything that, which means that the points that are above the 0 means that mineralization was occurring on that plot. Below 0 means that immobilization was occurring on that field. So first, I have only the yellow one is only the winter fallow, so the plots without CRI, and we can see that it's around 0, and some plots are doing mineralization, but when we add the red one is the results from the sampling with CRI. So this is the whole season. So the first one being our, I think at the planting, the day of planting, and the other one's pre-harvest. And we can see that throughout the season, due to rain and other factors, we have a great we have some variability on when it's mineralization or immobilization, or even we have a net 0 working happening. Sorry. So that's, we can see what's going on through the whole season, but our question was our results were net positive or negative? So we have a nitrogen mineralization on the y-axis, but now I have the different nitrogen rates on the x-axis, and on top I have the three different landscape positions. And we can see that overall, except for 2 treatments, we either have a 0 net or a positive net. So most of the time, either nothing or mineralization was occurring on that field, except for the 2 high nitrogen rates on the summit, on the summit and the toes low position with CRI. So we had 1 dry and 1 wet year on this research. So we're hoping with a 3rd year, we may get a more, a 3rd year to validate our data. I don't know what's the right wording for it. And I have on the next slide, I have some yield data, which that was the focus on our for research, was the yield drag. So as you can see, CRI, sorry, again, we have the nitrogen rate on the x-axis, and now we have yield on the y-axis, and the 3 different landscape positions on top. And and we can see even in the different landscape positions, the plots with CRI yield less than the plots without CRI. And with the colorful dashed line, we can see the agronomy the AONR, the agricultural agronomy. Agronomic, sorry, the agronomical optimum nitrogen rate for both of CRI treatments. So the AONR for the CRI treatment is 145 kilograms per hectare. It's a little bit cut over there. And the plot with the winter fallow or without CRI, the AONR is 156 kilograms per hectare of nitrogen. So the conclusion of my research is that CRI has an effect on mineralization, on the mineralization dynamics in the cornfield, especially on the high nitrogen rates. It was possible to see a yield decrease on the plots with CRI, but even though we have the AONR, I think some economical optimum nitrogen rate would be necessary for these plots. Would be nice to see like what would be the economical threshold. And my 3rd conclusion, as always, we need further research to understand the effect of CRI on corn yield, and we actually already are working on the second project that hopefully is gonna answer more questions about it. So with that, I want to thank the INRC for funding this project, the McDaniel and Lichty Labs for all the help on field work and computer work. And I want to thank all of you for watching this presentation, and if you have any questions, I'm here to answer.
There was one question online that I'm gonna ask first, because I think it might clarify something.
Okay.
Are these experiments following soybean or corn?
So the CRI is planted after soybeans. So that's why we change it fielding every year.
Any questions from the audience?
Yes.
When was the zero right terminated?
So the CRI, forgot to mention, the CRI was terminated 1 or 2 days before planting corn because we want the maximum biomass possible.
Did you measure the biomass?
Yes, so I have the measurement of the biomass.
Cool.
I have the CN ratio analysis on that, but I didn't have time to work on that data yet.
Safe to say that it's less than 2,000 pounds even.
And for the CRI biomass, we saw an effect on landscape position on the amount of biomass. So the toe slope produced more biomass than the summit and the back slope.
How's it look this spring?
Same.
Yeah.
Yeah.
Does it look pretty? Have you gotten a lot of growth so far this spring? Have you been able to go out
Uh,
and see where the plots are?
I took the first sampling when, so we do one sampling at the, when it warms up, when the end of the dormancy period. Yeah, it's like this tall, so it's a little bit bigger than what it was last year, so I'm pretty pleased with it.
Okay, I have a question. This is for you and Dr. Lick. What did you define as the end of dormancy period this year?
January 1.
Yeah, but-
Oh, that's a great question.
Because it got nice, but then it got cold, yeah.
Well, I waited until we had a consistent few days. So we took our sampling on that week that was was like first few days of April. So we waited until it was consistently warm. Yeah. But I don't know if Dr. Mark has another.
Anyone has a suggestion on how to?
Yeah, so that would be.
Yeah, I'm all ears.
Usually you don't have a year like this where end of February, we all.
Yeah. We were,
Yeah, there's a lot of fields that were green.
yeah, we were ready to sample in February and in March, and then it snowed again, so.
I would think most years, not this year, could maybe go by fall.
Yeah. You can almost go by the calendar.
Yeah.
It's usually gonna be the last week of March, first week of April. But this year, yeah.
So we have one additional question online for Arturo. Did you try to correlate variation in time with variation in yield? Or did any time variable of nutrients correspond to yield?
No, I haven't really played with the yield data yet. I have it, but I haven't correlated with soil with any kind of analysis.
There's also a comment online. Excellent presentation, both of you.
Thank you.
Thank you.
Is that all for questions? Anybody else? Once again, I'm gonna thank Mark. You know, we can use it, thank you.
Well, you gotta do some work for those. That come cheap, eh? The fully wedged that arrive on time.